Just to make sure that everyone is aware, today's presentation is being recorded, and if you're uncomfortable with being part of that recording, we understand. For that fact, we'll be sending the replay out to all those that have registered, so you're welcome to watch the recording then. During the webinar, we'll be running a live Q&A at the end of the webinar. We may also stop for questions when that time occurs. Ben will ask for questions. And so for all those burning questions during the live Q&A, could we please use the raise a hand feature? Now, this is a feature that's at the bottom of the Zoom screen, and when clicked upon, you'll raise your hand, and I'll give you the option for that attendee to unmute themselves and ask a question directly to Ben. We've also enabled the Ask a Question feature, which is a great option that you can use to ask questions during and after the presentation. To use this feature, simply use a question and answer symbol at the bottom of the Zoom screen. That will open the Q&A tab, and just pop your questions in there. We'll answer these questions during the live Q&A sessions. Please feel free to also upvote questions if great minds think alike and you see a question that you really think would be great to ask. You'll also notice there's a chat feature, and if you need to communicate with Ben or myself during the presentation, please use this. It's also located at the bottom of the Zoom window. So for example, if there is a sound or an image issue, use this chat feature to directly communicate during. And if I missed anything or just want to share the presentation with colleagues afterwards, don't worry. As I said, we'll be sending around the on-demand recording when it's available. So today it's my great pleasure to introduce the speaker, Dr. Ben Walters from Genentech in San Francisco. After completing a Bachelor of Science at North Carolina State University, Ben then undertook his PhD studies at the University of Pennsylvania under the mentorship of Professor Walter Englander, who was a pioneer in HTS mass spectrometry. During these studies, Ben developed an expertise in protein structural dynamics, protein folding, macular complexes, and advanced structural mass spectrometry techniques. After a successful PhD, Ben then began his scientific career with a prestigious postdoctoral fellowship at Genentech in San Francisco, joining the Department of Protein Analytical Chemistry. During his time at the biotech company, Ben has moved into a variety of positions from early stage pharmaceutical development, biochemical and cellular pharmacology, and is now in an independent research arm called GREDI. Ben now heads up a strong research group at Genentech, who plays a central role in early stage research and discovery efforts, specializing in the identification of lead drug candidates by developing customized high throughput screening applications, and then utilizing technologies such as hydrogen deuterium exchange mass spectrometry to characterize the mechanism of actions of the lead candidates once they're identified. During the early days of his career, if he wasn't found focusing on protein folding and dynamics, he would be found out in the San Francisco Bay, flexing his sailor muscles with his boating crew. Still an avid sailor today, however, these days, he and his wife are looking to the future with their new family crew, their 3 beautiful children, with the newest member I'd like to add was also born last month. So really excited for this presentation and looking forward to a great talk, Ben. Thanks.
Thank you. All right, well, I guess I'll just take it from here if you can hear me all right.
Yep.
All right. Let's see. Okay, so I guess the title of my presentation today is Running the Gamut, Tales of Protein Dynamics and Drug Discovery. I thought I'd just select a few stories for you from my career so far here at Genentech, which has actually been quite diverse. I've got this map here on the left that kind of shows the whole range of different applications that I guess we've kind of stumbled across, mainly in the form of people coming into my office with interesting protein dynamics questions, and then one thing leads to another, and we end up, looking at it by hydrogen exchange or some other technique. So I guess to get things started, maybe as a little bit of a warm up, I thought I'd ask you guys a question. So some things that I like, beer, of course, and hydrogen bonds. And this guy right here in the middle, Kai Linderstrom Lang. I don't know if many of you know what these folks have in common. This guy's genius actually extends miles, because he was able to get the friendly folks over in Carlsberg Labs to pay him somehow to study proteins and protein dynamics. We're very fortunate that he did that, because This was actually, he was getting into this field before protein structures had even really been solved. And he somehow had this foresight to know that proteins are actually these dynamic entities that are constantly folding and unfolding, and that their activity is kind of born through these motions that they have. This is a quote that he said that always kind of impresses me, given the fact that he said this back in the 60s, before we knew much at all about proteins, aside from the fact that they had secondary structures and hydrogen bonds were probably important. Before I get too deep into this presentation, I thought I'd at least mention that there's some interesting collection of seminars that we're holding with the International Society for Hydrogen Deuterium Exchange. If you're interested in this, please go to the website shown here, hdxms.net seminars, and check this out. Hopefully you'll enjoy it. Trajan's actually one of the sponsors of these seminars too. So I figured I would get started by telling you kind of how I view amide hydrogen exchange, how it works, and how I like to plot it or think about the changes that I'm seeing. And then try to give you an overview of a few things that I've done with it since joining this early discovery group over in Genentech. And actually some of these projects, have spanned many years. And it's been a pleasure to see, how the molecules changed over time. That's especially the case with tryptase. Hopefully I'll also be able to kind of touch on some of the things about hydrogen exchange that I appreciate. Mainly how dynamics really, to me, bridges the gap between, a static structure and then the activity that structure does. And I think that working together with structural biology, the physics of protein motion, we can really start to uncover some structure-function relationships that we're seeing in biology. So I'll just jump into kind of some fundamentals of hydrogen exchange. It's actually not very complicated. Sometimes interpreting the data can get a little bit complicated, but so this little diagram here was actually written down back in the 50s, and it's still kind of how we like to think about hydrogen exchange theoretically. That hydrogen bonds are basically protecting protons from exchanging with water, as long as they're formed with self. And we can describe the opening and closing rates here with some simple kinetic rate constants. And then once the hydrogen bond is broken, basically because hydrogen bonds are so important and so valuable in terms of energy, they're either always formed to self or formed to water. And so we know that once the intramolecular hydrogen bond is broken, that it will be formed with water. And all of a sudden the rules will be governed under basically chemistry that's well understood under this chemical exchange rate. So I guess kind of the overall idea here is that the H bonds are going to protect the proton from exchange, and it's going to sequester it. So that period of time where the H bond is established, the proton can't exchange, then the other period of time, we know how it's going to go. And so with this, we can actually back out information about the opening and closing rates here. We can control this reaction very delicately, actually, in the case that the H bond is broken by things like adjustments of the pH or the temperature. The donor pKa is actually the thing that determines the sequence dependence of hydrogen exchange. This is actually coming from neighboring inductive effects. We can't really control that, but we understand the basic rules that govern these rate constants. And it actually works out really nice for us because we can do experiments at physiological pHs, over here on this side of the plot. And here, we have exchange going extremely fast. Up at pH 9, we're talking in microseconds, milliseconds. The proton will have completely exchanged in all of the molecules in your population. And then we can drop the pH down to some low value like 2.5, and this will give us time to actually look at what's happened before we lose this information due to a process called back exchange. To kind of give you a flavor of what this experiment looks like with mass spec, because you may be wondering, well, how do we do this by mass measurements? So basically we'll take our protein, we'll dilute it into D2O. In this case, we'll start with a fully proteated protein, so it's got protons H on it. And then in time, the protein starts exchanging. This is dependent on its structural stability, et cetera. And then we can take samples as the experiment's ongoing, and we can try to slow down the exchange by doing things like dropping the temperature, bringing the pH to 2.5, and then we can digest the protein and separate it on a column. Now that exchange is going very slow, as long as we move fast enough, we can actually preserve most of this information. And so then what you're seeing over here on the right-hand side are multiple overlapping peptides that are going into a protein structure starting from the N-terminus. The dashed line is the chemical exchange rate. And essentially what we're seeing is that something interesting is going on, say, at about residue 11. And then as we start marching into the protein structure, we're getting more and more and more residues on these peptides, or exchange sites, if you will, exchanging and in this protected state, or in this slowed state. And I guess right now I should say that the reason I tend to focus on amides opposed to all of the other protons out there is because all of the other ones either go too slow or too fast to measure. Things on carbon are exchanging at rates that would take you, thousands of years to measure. Things like alcohols are exchanging so quickly that you would never be able to trap that in order to make the measurement. And so it works out really nice that this hydrogen bond, which is the most ubiquitous thing probably about protein structure. Structured proteins have hydrogen bonds with self on their backbone amides. And so by studying these, we can actually learn about protein dynamics at the individual residue level. So spending a little bit of time on theory, and this slide will go by hopefully quickly for those of you who just want to kind of think about plots and whatnot. But I like to think about, so if this is kind of a theoretical exchange trace, let's say, for a particular peptide, and it could take any of these different values that, the blue line, the cyan line, the green. And what would this mean compared to, say, the chemical exchange rate that I talked about, as it depends on pH and temperature? Well, there are these expressions that you can write down, and they've kind of stood the test of time. We've been using them now for more than half a century, because they work out really nicely to tell us some things about interesting quantities that we care about with proteins. For example, the free energy of unfolding. We can extract this depending on the conditions. If the conditions are right, if the closing rate is, say, a lot faster than our chemical exchange rate, and that closing rate is a lot faster than our opening rate, which tends to be true for folded proteins, we can start to back out quantities that are biophysically relevant. And regardless of how we want to look at it, we're always going to be measuring essentially a sum of first order exchange rates to describe these curves. So regardless of what the protection factor means, in terms of whether it means the opening rate or whether it means, say, the KEX rate, which is shown up here. So this is like we put a stable protein into D2O and it starts exchanging at some rate constant. It doesn't quite matter which one of these is operative for the sake of how I like to think about changes that I'm measuring in the experiment. I like to think about them in terms of, in this case, offsets from the chemical exchange rate. So if I'm just looking at a single protein in solution, or in experiments that have become very popular these days because of how difficult it is to solve the structures of some proteins, epitope mapping experiments have shown to be incredibly popular with hydrogen exchange, because basically you're now comparing two states. Instead of comparing a chemical exchange to a particular thing that I'm measuring in an experiment, I'll be comparing two traces that I've measured in an experiment. I'll be looking at the changes. And if I want to talk about things in terms of a protection factor, like this arrow is showing, it's kind of nice to look at this stuff on a log plot, because I can say, okay, the change is 10 because I move a decade here between 10 to the 1, 10 to the 2, or the change is 2 because I've moved 2 decades. It's really all about how long did it take to see that same amount of exchange in your two different states. So the experiment's actually really easy to kind of think about when you look at it on this level. And at least I hope now you could appreciate that there's some theory underneath these traces, that this is more than just me kind of drawing on them with arrows. We can actually look at these experiments and we can get this wealth of information by asking questions, well, where are the differences? Where in the structure? Because remember, I can measure this, for each individual amino acid in the protein, with the exception of proline. And so I can tell, well, this region of the molecule was affected, or that region was affected. In a sense, you can think about it, and in this case, I'm showing you information that's coming from a single molecule, looking at its stabilities. In a sense, you can think about it as the structural biology is providing this base information, which is necessary because hydrogen exchange is not a structural technique. I can't necessarily say this is a beta sheet, that's a helix. These two things are interacting for sure. But with the information from structure, we can really start to say some really interesting things about structure function. And then this kind of represents a dream, which is coming true right now. And that's using hydrogen exchange and molecular dynamics together. So I'll say one quick word about that, in case any of you out there are doing some molecular dynamics and thinking that that's a pretty crazy thing to say. Well, we're getting pretty good at simulating extended periods of time. Right now, we could go for easily 10 microseconds in a week, let's say. So getting us into this area. And the main takeaway from this plot is that depending on what the protection factors are, and you can see an idea of what protection factors from HXMS look like on the right-hand side. Depending on what the protection factor is of a particular proton, I'm going to have to wait a certain amount of time until I see it. And when we could only do simulations that lasted, say, 20 nanoseconds, we didn't have access to very much of this space. But as we're getting into longer and longer simulations, I think we're going to start to see some real interactions between computational biophysics and what we can measure with hydrogen exchange. So without further ado, I'll jump into some of the stories and get a little bit more concrete and kind of get a little bit away from theory. Some of my favorite stories for a number of reasons. Not the first because I've tried to talk about it at least 6 or 7 times, and the legal department said no. But finally, the team was able to move the molecule into development, which then kind of unlocked our ability to publish some of this information. Henry Mahn, who's shown here in the picture, was the lead of most of these experiments. It began when he came into my office, actually, as a postdoc, when I was a postdoc at Genentech. And we've been doing work with tryptase now for, I guess the last 5 or 6 years. And some really interesting things have come out of it. So I'll tell you a little bit about tryptase. I'm not going to spend too much time talking about the biology. Suffice it to say, tryptase is a relevant target for pharmaceuticals because it's the primary driver of a particular type of asthma. That's not kind of the area of my expertise, and so I'll just focus on kind of what my role was in this process, and really it's to kind of understand what happens with tryptase, how does tryptase activity work, and what might be ways that we would be able to inhibit this beast. So you might be familiar with trypsin, and trypsin is like tryptase in the sense that it's produced in an inactive state, where there's a signal peptide and a propeptide and then a mature enzyme. And this video is showing you the conformational changes that have to occur upon clipping of the pro domain. This leads to activation of the molecule. And this family of molecules are very similar. Tryptase is a little bit different than trypsin in the sense that in addition to requiring this cleavage event and these conformational changes, will also depend on tetramerization or tetramer formation. The tetramer forms as kind of alternating monomers as you kind of walk around this cluster here. And you can see the different subunits here, A, B, C, and D. And basically, the way to think about it is that there's going to be a large interface here and a small interface. And tryptase has some extra residues here that are going to need to insert into its neighbor in order to be stabilized and in order to become catalytically active. And then basically it can start to chew up its substrate as the substrate passes through this channel. And so we want to inhibit this activity. And this was the plot that Henry came into my office with to kind of get me interested in tryptase, because we couldn't get a structure. The problem with tryptase was that if you tried to structure the single monomer, or if you tried to solve the structure or generate a crystal of the single monomer here, you just could never get the crystal to form. And if you solved it in the state of the tetramer, well, it's active, and so most of the antibodies that you're going to pull up that are interesting are going to push this protein out of its active conformation into the inactive state, which then is going to prevent you from getting the crystal. But there's some really interesting observations. All of these antibodies were observed to compete with one another, and so they shared the same epitopes. However, if you looked at, say, the one 31A.V11, we'll just call that 31A, it works as both a Fab and an IgG. And that's interesting because a Fab is basically, if we took the standard shape of an IgG, and it's kind of the classic Y shape. It has two Fabs on either side, connected by this hinge region, and then this FC, or fragment crystallizable domain. If we chopped off the Fabs, then we'd have these little individual pieces floating around. And actually, the antibodies always bind to their targets through these complement determining regions, which are at the tips of the Fabs. And so the question is here, well, why does 31A work as a Fab and as an IgG? Whereas E104 versions 1 and 2, they require the full length IgG. So there must be some sort of a difference in their mechanisms of action. Also, there's this really interesting thing called the hook effect, where one of these antibodies, 104.1, basically at higher concentrations stops inhibiting tryptase. Well, without any structural information, we're kind of shooting in the dark here, or at least the project team was. And it was even more fascinating to me that there's only two residues different between 104V1 and V2. And these residues are not in the complement determining region. So these are in the constant domains of the antibody. And so, it's, I guess, not too uncommon that we go into the lab and we see something that we didn't expect at all and kind of lack any explanation for. But here it's just kind of a number of things are really interesting. Well, how could the epitopes be that different that they don't? So, we had lots of questions that we needed to address. I was getting ahead of my slides a little bit looking at that last one. So here the highlighted regions are showing epitopes that we found by HX. And this is the way that I showed it at the time, looking at just the things that happened on the surface as a way to kind of get through some of the allosteric changes that we see, which I'll talk about in a few more minutes. But we could clearly see that they shared the same epitopes, and we were left asking these questions. Well, why does 31 work as the full length molecule and the Fab? Why does E104 require the tetramer? And then how can we use this information that we've gotten by HX to maybe suggest a path forward for crystallization? Well, we were starting to think at the time, and actually just as we had this data collected, Henry and I were kind of sitting in the conference room and wondering, well, it kind of looks like that the epitopes shifted a little bit here. So maybe there's something about the hinge and something about the distances here between the binding interfaces. Maybe it's that two Fabs can fit at the same time on either side. So you'd have two kind of sticking down to the bottom and then two sticking up to the top. But maybe in the case of 31A, that wouldn't be possible. Maybe the Fabs would kind of bump into each other. So that was kind of in the back of our minds. All the while, Henry had been trying lots of different ways to crystallize this 31A bound to the single monomer, like you see here. So now I'm kind of showing a little bit more of the HX changes that we saw in 31A. And it occurred to Henry as he was looking at it that, well, if we were stabilizing these regions by binding to the monomer, and if our binding frame was up here, maybe one of the active site inhibitors that exists for other members of this class of proteins would work. Maybe they would actually calm down some of the motions that are becoming problematic and basically preventing us from getting a crystal. That's what these are going to suggest here. Was that maybe an active site inhibitor will fit. Now, usually making the complex larger is bad for X-ray crystallography, but Henry thought that maybe it was worth a try because, well, what else can we do? We basically run out of options. He'd actually tried a lot of different active site binders for this protein unsuccessfully, and so these were Henry's words, that he never would have tried adding this active site or this soybean trypsin inhibitor into this mixture if it wasn't for the hydrogen exchange data. But in fact, doing so allowed a high resolution crystal structure to form, or a high resolution, a well diffracting crystal to form that then produced this high resolution structure that you see. And so anyways, it was really interesting how hydrogen exchange was able to factor into the mix here and really kind of help figure out how to get to this point. I thought at this point I'd step in and show you kind of like how the hydrogen exchange protection factors can be used to think about what the binding frame might be. Because probably many of you are familiar with the fact that when you do a hydrogen exchange experiment, you see everything, not only things that happened at an interface, but you also see any downstream effects of those changes. So here I'm showing you changes in protection factors versus the tryptase sequence, and putting basically where we saw the crystal contacts, which you can kind of see on the left, onto this figure. And what you can kind of see is that your most intense protection factors are the ones that are delineating the binding interface. And these lower level energy signatures are the downstream effects. That kind of makes sense because you can't transmit information with perfect efficiency. If you could bind and then transmit that information with the equivalent energetics throughout the molecule, well, that would be perfect efficiency. And so you can kind of use these rules to deduce that the highest intensity protection factors must be where the binding interfaces are formed. And it's interesting to consider that, well, I don't know how much hydrogen exchange data you've looked at with respect to peptides, but a lot of folks are really focused on the difference in deuterium uptake. But really, I'm interested in the time difference between when the level of deuterium uptake is the same between two conditions. And in our largest protection factors here, basically, there's not even any overlap. We haven't even gotten to that point in this particular experiment. And so I've actually written some computer programs that process this data. And this is actually set as a limit in this experiment. I barely was able to measure this value of an energy change in one peptide with the uncertainty that's shaded here on the bar plot. And so I'll extend that energy to anything like this, where there isn't even an overlap in time. But then these things that are smaller in energy, they are because where we have the unbound state in black and the bound state in red, I'm actually able to do this time comparison. And so this is basically how I get to these values. These protection factors can be thought of as like unit areas between the two states. And they're very useful for suggesting where direct interactions have occurred. Going back to just how can we use that crystal structure that we just got, and then also this earlier hypothesis about why does 31A work as an individual Fab, but E104 does not work unless it's in the full-length antibody. Well, what you can clearly see now, if you model the crystal structures that we were just looking at with the tetramer, the hypothetical tetramer, is that there would be all of these clashes between adjacent Fabs. So this basically tells us that the mechanism of action for 31A was because it's binding to monomers and preventing this tetramer from forming. Now, there might be a little bit more to it, but at least at this point, it was interesting that we were almost able to see that these antibodies had different mechanisms of action just by looking at the exchange data. And this was kind of our natural hypothesis after we looked at those exchange patches earlier, that you couldn't fit side by side. All the while we were looking at that data and time was passing, we actually got our first cryo-EM structure at Genentech. This was really neat. I was working with the guy who's leading that group now. And so his very first project when he came to Genentech was, can you help us figure out tryptase? And we were kind of working on it at the same time. And so when he came back with this 15 angstrom resolution structure, and this is a little bit easier to see than looking at energies, you can see, how close the regions that I was able to annotate by hydrogen exchange are to the hypothetical interface. Another interesting thing to note here is that both fabs appear to be on a tetramer at the same time. And so that was kind of what we thought might be the case. That was why we were thinking, perhaps E104 doesn't work as a fab, because maybe that small difference in the epitope or the binding interface was just enough, and so that all of the fabs could load up this tetramer without causing dissociation. To kind of double back on that and do one more experiment to show that this probably is the case in terms of the mechanism of action, we were thinking about, well, what could we do to kind of interfere with the hinge to show that we could basically turn a full-length IgG into a non-functional one, or one that would not inhibit tryptase. And so the idea here was that, well, if we elongate the hinge a little bit, maybe we can, abrogate this effect where the tryptase is, where the antibody is either binding to tryptase in a strained state, and then in relaxing, it's causing the dissociation, or it's binding selectively to the monomers. One way or the other, this mechanism seems to be operative. And so maybe if we add some amino acids into the hinge, we'll be able to see this. Can make that point a little bit clearer here, and this Easter Island man is just being used to kind of show you the difference in orientations between everything that we're looking at here. If we look at a model of the fabs and a hypothetical FC fragment taken from another crystal structure, and we consider, could the hinge region span or reach across to touch two fabs that are bound to the same interface? Well, you can kind of see from these fiducial markers that basically these two things would need to line up. You would need to have the C terminus from this fab overlapping with, the N terminus from this fab overlapping with the C terminus from this one here. And I apologize if I'm mixing up the directions, but I think you get the idea that basically they need to connect, and they're 12.6 angstroms apart. So that's a pretty sizable distance. That's more than 20% of the distance, say, between two fabs and your standard IgG framework. Here we're actually looking down. So we've taken this structure and we've rotated it and looked down. And so you can imagine the FC is sticking out of the page. And this would be about the expected distance, but it needs to go, another 12 angstroms in that direction and another 12 angstroms in that direction. And so the experiment that was performed was, well, just simply let's add some residues here. Basically, then we'll be able to span this gap, and then should not see any activity if this hypothesis is correct. Just kind of as a quick aside, it's important that we didn't lose affinity for tryptase. We still want to be bound to tryptase in these experiments. And as it turns out, just like Henry was suspecting, and Henry really drove most of these experiments, in these extended hinge mutants, you can see that we lost activity. And the longest hinge version, and they're roughly in the order that's shown down here, but you can kind of see from the way that these notations have been made. In the longest hinge mutant, we were basically looking at what we saw in the case of the fab. And as it turns out, we never were able to get a crystal structure with version 2. And so we're left to wonder exactly what was going on with the hook effect that we saw earlier. But we think it's probably has something to do with the way that the fabs are engaging each individual monomer of the tetramer. And I thought it was also really interesting. So this was a map that I made just by looking at these protection factors and using overlapping peptides to narrow down regions of change, which there are differing opinions in the field about how to use overlapping peptides. Personally, I think getting information that helps us move forward is really the goal. And I was pleased to see that, you know, these changes really did line up. I mean, at a very detailed level, aside from W38 here, which I did not see in version 1. And remember, version 2 is not what we're seeing in the crystal structure. There were some huge, I mean, there were some incredible similarities, you know, 47 to 50, and then we have a contact at Q50. Or this is just the notation or the way that we talked about the tryptase sequence. But it was basically spot on here. I mean, well, you know, using the highest intensity protection factors, you could say what was directly interacting. And then there were also these downstream effects in the case of E104. And we think that maybe some of these are responsible for why we don't see a hook effect in V2, and we do see a hook effect in V1. In any case, it's fascinating that simply 2 amino acid change away from the region that's forming the protein-protein interface in the antibody is eliciting changes that I can measure by HX in the target, right? I mean, that's incredibly removed. And it's really interesting that you're actually able to see that. So, you know, I think that this kind of demonstrates the power of HX when used in combination with other methods, is that we're able to identify differences between the versions of E104. We're able to kind of create some hypotheses about mechanisms of action and test them in the lab and see that they're true. Working alongside of all of these other technologies, you know, there was kind of a really neat synergy that happened. I think that the hydrogen exchange was remarkable in the fact that it agreed with the other structural methods. And well, it took a lot less time. To be specific, it took 3 days. It took 3 days to do an individual epitope that's being shown here, and about 4 hours to analyze it with some of the software that I've got. And so it really did take a lot less time than some of these other structural methods. Now, we still relied on that information, but it's just interesting how they can all work together. Excuse me for needing to clear my throat. All right. So if there are any questions about tryptase, I could take them, or we can go into the next story about IRE1. So IRE1 is an interest. You have anything, Robert?
Go for it. I think there's a really good question from Tom, but I think we might hold that to the end. So continue on.
Yep.
Yep.
And so the next story I'll tell you about is now moving to the small molecule world. You know, a lot of us are familiar with doing hydrogen exchange in the context of large proteins or large protein-protein interfaces. But what about small molecules that are only interacting with a few amino acids? So IRE1 is going to be a story where we're going to look at small molecules interacting with a protein target. But I'd be remiss if I didn't tell you a little bit about IRE1, because it's a fascinating protein. So what you're seeing here is actually the cytoplasmic domain, and I'll tell you a little bit more about what's going on with its activity on the next slide. But it's an interesting protein because it has a kinase domain, it has an RNase domain. The kinase domain basically is solely responsible for trans-autophosphorylation of itself after it dimerizes, which then stabilizes the protein, communicating through some allosteric changes to the RNase domain, that the RNase domain now can become active. It's responsible for a lot of things, and so there's great interest in how to modulate it, and plus there's a lot of ways to modulate it with the kinase domain and an RNase domain. To kind of look at maybe one of the reasons why people are intrigued with IRE1 and why it's such an important target, and also why we want to generate small molecules to kind of be able to push it around. That's how we can learn more about it. You know, so we need to be able to generate things that bind to it, that have some sort of activity when they are bound to it. And then we need to kind of be able to understand that activity in order to really dig into the question. Well, let's just get this started. So once you have, so basically you have a luminal domain in the ER, and it's going to participate in the unfolded protein response. When enough, let's say, unfolded protein is there, it will dimerize with self. And it's not limited to this dimer, but when the dimer forms, and there are multiple kinds of dimers that can form, you can basically dimerize this kinase domain and get transphosphorylation at 3 specific sites. This process is highly orchestrated structurally, but that's not the area where I've focused. And so I'm going to leave that to other people so that I don't just completely botch it. But as far as I'm concerned, the process of bringing the kinase together leads to transphosphorylation and then a reorganization into a back-to-back dimer. This back-to-back dimer can then activate the ribonuclease and to XBP1 transcript activation, which then basically leads to a transcription factor, which stimulates the production of proteins that then help to relieve some of the stress on the ER. But it doesn't stop there. It can reorganize. This is actually one of the reasons why this protein can be frustrating to work with as well. It's not limited to a dimer. When it becomes active, it can hop into these higher level oligomeric states. And not just one, there are multiple types of oligomers. And we don't understand these oligomers very well. They do lead to activation of the RNase domain. And these things can be clearly shown through molecular biology tools to be important. They actually are doing something called regulated IRE dependent decay. This is not very well understood, and it's operated in a wide range of different diseases. But one thing that does seem kind of necessary is this back-to-back dimer. So, everything upstream of this step seems kind of necessary. It also seems like, well, you've got to get these transphosphorylation events. But what if we could maybe activate the kinase through a kinase modulator? Could we do something that way? And it turns out that yes, you can actually, there are kinase modulators that activate the kinase, and they tend to push for this back-to-back dimer without phosphorylation, of course, but they will activate the RNase domain in some cases. And then there are other cases where you have inhibitors of the kinase that will basically not result in your back-to-back dimer. And in that case, you don't have your inactive ribonuclease. So pretty straightforward, until Elena found this to be the case, that you can have kinase modulators that don't promote the back-to-back dimer that do activate the ribonuclease. How can that be? And if that question is interesting, the next question just really is fascinating. Here, these molecules shaded down here in gray are the two suspects where 1749 here is basically looks identical to an inhibitor that was already on the market, a kinase domain inhibitor that was already on the market, AMG 18. And sorry, ribonuclease domain inhibitor. So this is an inhibitor of RNase activity. All of these are going to inhibit the kinase domain activity, but we're going to be focused on RNase activity. It's easy to get them mixed up like I just did, actually. But it's fascinating that you have an activator over here and an inhibitor over here, and the only difference is this small change. So, okay, well, what could be going on there is, I guess this tail in 1749 must be driving dimerization, whereas if you don't have that tail, you're, say, not driving dimerization, right? Because dimerization is how we're going to get our activity in the RNase domain. Well, as it turned out in AUC experiments that Elena was doing, what she was seeing, and very similarly to what the phosphorylated protein looks like, it starts to get heavier, and that's because it's dimerizing in these analytical ultracentrifugation traces. But over here, when you look at say 9807, for example, it's an activator and it's behaving like phosphorylation. It's activating the ribonuclease domain without without having an active kinase domain. But yet 1789 is just this outlier. It's giving us some activity over here, and it's not requiring the back-to-back dimer. So it seems like the mechanism that 1789 is using to activate the RNase is a different mechanism than the one that 9807 is using. And it's also just kind of fascinating. In this case, our goal was to find activators of the RNase domain. And the reason initially that Elena thought that this one was interesting was because she knew about this molecule. And her screen randomly pulled this one out. But it was kind of an interesting setup, Why are these changes, why are these small changes leading to such remarkable effects that we can observe by a number of different assays? And I should say before jumping too much further forward that in the cell, we don't think 1789 is dissociating the dimer, but we do think that its mechanism is independent. There's a really nice manuscript that was on the earlier slide that Elena wrote summarizing all of this data. I encourage you to check that out if you're interested in learning a little bit more about this story. But I'll just kind of touch on a couple of the highlights as I'm moving through this area of the talk, which was that if you looked at the crystal structures of 1749 and AMG 18, this really didn't help you understand what was going on. Why was one compound leading to activation and the other leading to inhibition? And I guess that the thing to note here, and there's a lot more going on in these pictures, but that the main changes that we see are occurring between these residues, 711, 712, 612, and 599. The reason that's interesting is because, whoops, wrong direction. Those weren't really the residues that we saw to be, really interestingly affected by hydrogen exchange. And the residues that we did see to be remarkably affected and seemingly fit the picture of what we were seeing everywhere else was the activation loop. And the activation loop doesn't show up in crystal structures. So we don't really know what the activation loop is doing in this protein. But you can see that 17, well, I should say we do know that the activation loop is critical, but we don't know why these changes in dynamics, or we didn't know why these changes in dynamics are so efficacious towards getting activity in the RNase domain. When you drive towards your dimer, you're stabilizing the activation loop. And you see this in the phosphorylated protein. You also see this in 9807. But in 1749, it's destabilizing this activation loop even further. And AMG 18 is basically doing nothing to it. So it's preventing it from looking like the phosphorylated state. And prior to this, that's kind of how we thought things worked. If you forced it to look more like the 3P state, that's how you got activity. This is just cataloging the significance of the effect in log protection factors. These are not tiny changes. These are pretty large changes in energy that are going on in this loop region. And when I color the global changes onto the structure that I was just showing you, and here we've got one of the monomers. And the way that I'm showing all of the hydrogen exchange data here is kind of by showing areas that were destabilized by compound binding in red. And there's a gradation, and so things that were the most destabilized are dark red, and they're fat on the structure here. And then things that were stabilized by compound binding are shown in blue, and the tube gets skinnier. What you can see is that, well, 9807 stabilized the kinase domain. That makes sense. That's what 3P does. Phosphorylation stabilizes the kinase domain and allows for these higher level, say, dimer and oligomers to form. So that kind of fits exactly with what we're thinking. And so looking over here at 1749, we're seeing something very similar to what we see in AMG 18, which isn't too much of a surprise in the sense that we know AMG 18 kind of, well, at least it doesn't promote dimerization. And that's through the C helix here. It's destabilized the C helix, which tends to be thought of as being important in forming the dimer. But the really unique thing that we see is that in an area where we're not even seeing where the chain goes in these crystal structures, we can see with hydrogen exchange that we have a huge difference. And now, I mean, this could just be hand waves. What good is this? I mean, it's just, okay, it's a difference. How do you know it matters? Well, I was just telling you about the small changes in the X-ray structure, right? When those compounds were bound, we saw changes in these residues. And this story is endlessly fascinating because, as it would turn out, 1749 does the opposite to the phosphorylated state that it does to the unphosphorylated state. And so that's not the first time we've been surprised by this molecule, meaning that it completely inhibits what would be active in any other case. It completely inhibits the RNase domain. But what's really fascinating is that when you make mutations of residues that were directly interacting with 1749 in the crystal structure, it's not having much of an effect on the differential, let's say. So if those changes were causative in what we're seeing with 1749, you would think that making a mutation should give you something in these experiments. But in fact, what we're seeing is that we don't see much of a difference until we start making mutants in the activation loop. When we start making mutants in this activation loop here, 687, now these will start to combine with areas that we saw in the crystal structure to give us very large effects. In this case, going back to enhancing the activity in the 3p state. There's a lot left to learn about IRE1, but I think, in a basic research setting, and then also in trying to understand mechanisms, I mean, hydrogen exchange can be invaluable to these efforts. And so, I think, we see that the activation loops dynamics are incredibly important, and that these changes correlated with the different activities. We saw that the mutations in the activation loop could abrogate the activity of 1749, which seems to suggest that these changes we're observing by HX are in fact important. And we're left with this question: well, why do kinase activation loop dynamics matter? And I think we have a new question, so that's progress. So just to kind of hop through this last bit, I think we're left asking questions. Well, why didn't we see the answer that we were expecting? And I don't want to run out of time here, and so I'm just going to kind of jump through these animations and tell you, well, perhaps we missed the difference. Hydrogen exchange activities are occurring over a huge array of timescales, and you have to wait for the native unfolding event before you're going to be able to see a modulation of the native unfolding event. In most cases, you're stabilizing a protein when you bind to it, and so you're slowing down the exchange rate. I mean, I like to think of it as you don't know what you don't know. And so in these cases, like this peptide here, all of my data is like a flat line. Or similarly, up here, I haven't really seen much exchange. Maybe the way that this change is communicated is in some of the space that I didn't measure. And so, just to point out, there are ways to get into longer time domains. I think Sheena, who gave the last seminar here, mentioned that she samples pretty far out into time, and so do I, because each peptide that I'm tracking in the HX experiment has multiple exchange sites, and it's important to recognize that what I'm seeing in an exchange trace down here is not the same thing. It's not the same sites. This is fundamentally new information that's happening up here, and maybe it seems a little bit counterintuitive because compounds can bind in a millisecond, but then we might have to wait weeks to see them. To see the effects in some cases, that would be even further out than what I'm showing here, but days are reasonable. I mean, this is effectively a week time scale. I've used pH modulation to get here, but I don't think it's too counterintuitive actually when you think about it actually in terms of something like sailing. You have very fast boats and you have very slow boats. If you say added an amount of drag, a fixed amount of drag to either one, well, if all you could see was the finish line, you'd have to wait longer to see the effect of that drag on your slow boat as compared to this boat. And then another aside is I hope to see Australia in the next America's Cup, because we got to get the cup back from the Kiwis. Anyways, before I digress too much, I think it's just really interesting to see how the time scales factor into this experiment, because even though things are happening fast, sometimes you have to wait longer to see it. I'll quickly tell you about my experiment cube, and the way that I like to think about it is really, it's not just the sequence factor. It's not just covering all of the amino acids, but it's watching them exchange. It's actually seeing them exchange that's really important. And probably one of the missing links from our RNA story could be because we were missing those sites. It also could be because we needed to study this in the context of the dimer, and we couldn't do that because then it would aggregate, but that's neither here nor there. There are tools that you can use to get at some of these extended time scales. You can use quench flow, or you can use the robotic approach, which is basically what I'm doing in my lab for almost everything that you've seen so far, is using the Trajan platform plus pH modulation. And that allows me to access a time scale from milliseconds to months. And even though some protein structure depends on pH, I can see that, and I can still use this effectively. So I cite these two works here, actually. Steve, who's with Trajan, I think he might even be online over there, was maybe one of the first people to do this pH modulation. But the idea is that when I understand what the levels of deuterium are that are moving around on the peptide or with solvent, and I understand the theory of HX really well, I can collect these different pH experiments, and these protons, like I was saying, they're different protons. So I'm measuring a different proton at pH 5.6 than I am when I'm looking at this reaction at pH 8.6 over this time scale. But I can actually stitch them back together, and I don't need to know any rate constant information to get here. And in doing so, I'm able to capture much more of the uptake domain, which is really what we need in order to say something about our results. So I really need to be able to completely sample everything in order to make positive and negative statements in these experiments. But I'm not sure if we have time to go into the next story. I've got maybe 4 slides on this next story. It's pretty quick, but I could also stop here and we can take questions and just call it an evening if you want.
I'm happy to keep going. So if you want to go through this next section quickly, and then we'll jump straight into a Q&A.
Okay. Well, just real quick, I'll tell you that you don't have to measure for so long. I'm glad I get to jump into this section because right after I've come off my soapbox about measuring for weeks, I mean, that makes the experiment a little bit inaccessible if you have to do that every single time. And you don't necessarily, because if there is a specific question that you're asking, like in this case, a question is, I got a bunch of small compounds and they're going to bind to this target. I want to do a single time point experiment that can reliably assess whether or not the compound hits in a particular region. Well, I can basically find a peptide that's giving me a maximum effect, and I can find that one time point by doing a survey experiment. I can also use this same kind of approach to distinguish between allosteric changes and say, a specific binding. So I'll just show you now. That's kind of a different tack than the statement that I can do a single time point. We're going to need to do more than a single time point to distinguish between allosteric changes and say promiscuous or a second site of binding. But you don't necessarily have to do so much effort if you want to get some information here. So in the case of say allosteric or non-specific binding, here's this protein that I'm just going to wrap up on real quick called HPK. And one of the peptides here, 38 to 57, I think this was one of the direct peptides. I'm sorry. This 110 to 117 is directly in the interface. But these other peptides are potentially non-specific binding. The drug is really small, so it couldn't account for the coloring pattern that you see here. And so, what we can do actually is start to titrate away the compound, and we can repeat our HX experiment every time that we change the concentration of the compound. And it's really neat. You can actually titrate across the bound state, and you'll see all of your exchange traces will kind of uniformly move together between unbound, which is here in magenta, and bound, which is here in black. So it's a really unique way that you can go in if you really need to differentiate binding sites to detect whether or not a change is a second site or whether it's allosteric. And then, here, like I was saying, you can do also single time point experiments where, I'm seeing that this is my region of maximum difference between all of these different compounds that I'm showing. Well, I could just monitor that single time point, and then that would give me, something interesting. To give one last plug for why I think we should think about protection factors in hydrogen exchange experiments, because here, with this particular set of compounds, you're able to derive a relationship just with this one peptide, because this is the peptide that is directly interacting. You're able to derive a relationship between the affinity or the equilibrium constant for inhibition and the protection factor in the experiment. And so, I think there's a lot that we can do with this technology. You can work fast, and in working fast, you still have reduced ambiguity. You can increase the information content of screens. You can actually start to get structural information from large screens. And you can use this to allow you to, say, pursue the correct lead molecule or make the right changes on your medicinal chemistry team. So with that, I'd just like to say thank you to the different teams that I was able to work with for some of the data that I'm showing here. And you can find more information if you check out these publications here on at least the tryptase and IRE1 stories. And And with that, I can turn it over for questions.
Thanks very much, Ben. That was really exciting. Great talk. We'll start with the question that Thomas Neville put forward in the Q&A box. I think it's a big meaty question, to be honest, but I think it's one that I've often thought about. So you made a comment early on in the talk that, you know, protein structure alone, protein HDX is not alone able to predict protein folds. Do you think the advent of powerful AI algorithms like AlphaFold 2 may allow us to use HDX in combinations with these predicting structure algorithms?
I hope so. I hope so. So you know, well, I guess one answer is that's what I'm working on in a lot of ways. Like, that's one of the things that I'm really preoccupied with right now in my research is how can we use hydrogen exchange together with computational techniques? You know, I don't know a whole lot about, I guess maybe I'm more, I've been more involved with computational biophysics from the level of simulations and, you know, physics-driven simulations, which tend to kind of be a little bit separated from AI. And so I haven't really thought a whole lot about how to use hydrogen exchange with AI. But I think it would be really wonderful to consider ways to do that, because I think that, you know, this integrative approach is going to be very powerful in the future.
Yeah, I think that the integrative approach, exactly right, is the way of the future for a lot of this molecular modeling. You know, whether you're drawing on cryo, whether you're drawing on HDX, cross-linking mass spectrometry, and other biophysical techniques. I guess leading on to that, what advice would you give to someone who's contemplating, you know, you said a lot of people come through your office and that's how a lot of your projects get started. What advice would you give to someone that's, you know, in the back of their mind have thought about HDX but haven't quite crossed that chasm yet?
That I think that there's a lot of, I mean, I think that so you can, you know, I don't know if I hope that this was able to come through in some of the slides, but with all the complexities of hydrogen exchange, you can also kind of represent these results in very simple ways. There are really great software packages out there that, you know, I'm not a spokesman for any of those, but I think that let's call it the activation energy to get into hydrogen exchange these days can easily be, you know, achieved. I mean, I think the reproducibility aspects of doing the experiments by hand are challenging. I mean, it was something that we worked on a whole lot in grad school. And, you know, I think when Trajan would come into Walter's office and say, hey, you know, why don't you guys buy a robot? Walter would be like, well, why would I need to do that? And when I have these wonderful, energetic lab students here, I could just give them a little bit of food. And, you know, and so, but, you know, for me, I mean, I really rely on it. And I think that anyone else who can get access to one basically can be off to the races. I think it's good, though, to be careful, to be careful to not overinterpret your data. And then just to say one more thing about the AI question, I think the only way to make hydrogen exchange useful in the context of AI is going to be to get away from thinking about the results just as changes in deuterium uptake. I think you have to get back to the physics of why did you see those changes in deuterium uptake. And I don't think that just leaving the results at a delta D is going to be quite enough. But could be famous last words.
I think the functional and mechanistic understanding need to really be brought to the table when you're really looking at this data.
Yeah. Well, I mean, you could have a long peptide that you're seeing a difference of 2 Daltons, or a short peptide that you're seeing a difference of 2 Daltons. And depending on when you looked in time, that difference might be very fleeting or it might be persistent. And I think all of those things are really important into connecting what you're seeing in these experiments to the dynamics that are occurring in the context of the protein.
Yeah. So just another question from Tom. Are there any computational tools that can help us calculate protection factors, time dependency on exchange, etc., and help us zoom in and design better HDX experiments? You mentioned that you wrote some of your own coding. Maybe give us some insight into the software that you've been working with there.
Yeah, so I don't have, in terms of going from simulation to experimental design, I've kind of, so because the timescales don't always are just starting to overlap, let's say. I've kind of used it in more of a heuristic or qualitative fashion. Create rules that dictate something from simulations. For example, if you see, I had a case where I had an antibody that we thought the tips of the fab were separating from one another because of what we were seeing in a molecular dynamics simulation. And so we reasoned that if that was the case, that in exposing this hydrophobic surface area, you would be destabilizing the protein, and we should see a selective change in HX rates just in this region. And so then we can go into the lab, look at what are the exchange traces in this region of an antibody, and then pick our time regime to do the experiment. So yeah, in that way, we definitely kind of crossed the threshold. In terms of like software, I mean, I think that there's software that's already out there that will try to estimate a change in rate constants or try to estimate a change in protection factors. I'm not exactly sure which softwares include that and which ones do not. I've tended to just like to do it in Python myself. I think maybe that's more common in some of the people that are, I guess, coming from like the old school of NMR-HX, which was where I started doing things. You just kind of like to have, your hands on the data. But I don't think that that's the only way to do it. I don't know whether that fully answered the question.
I think it was a great question. Anyone else, please feel free to either raise your hand or put some questions in. We're happy to take a couple more. Maybe on that computational tools, do you have any comments about, deep diving into data in a way that you were speaking about, not just the differentiable peptides, how software is handling that in whether a throughput aspect or whether or not it's having to really just look at the data deeper yourself?
Yeah, I mean, I think, utilizing overlapping peptides is tricky. And the cases where I've done that, I call that an art. And it's reproducible in the same way that like an artist can repaint something. But it's hard to specify, and I've actually tried and working on this right now. And I think if I had one of the guys who's working with me right now, Alvin, he'd be able to answer your question even better, because I've tried to show him how to do this too. And we've been working on it. We don't do it in every case. We don't really go through the overlapping peptides. But that's where I want to spend my time, to be quite honest. I'd rather spend my time, really looking at the peptides and saying, okay, if I add these extra residues here, how did the exchange trace change? The thing that takes some getting used to is that different peptides back exchange to different degrees because they influence one another. And so you can't just say subtract peptides from one another. There has to be more, it's a bit more involved than that. There are softwares that will try to basically use these overlapping peptides, but I think that there is a pretty heavy burden experimentally on generating enough input for those methods to work. But I guess I found that the human element of sitting down and thinking about the data and looking at it can be really powerful. It's become harder to publish things that way.
I think anyone that's really sat down and looked through individual peptides really understands that that's where the beauty of the science is really in that.
And I think that there will be softwares that will basically do this manual thing. And I mean, I've thought a lot about it, and I mean, maybe I would have worked on it if I hadn't been working on all of the other stuff. It's definitely a place where I would love to see it become automated, and I'm thinking about ways to automate it.
Yeah. I mean, fragmentation as well is also another step in the direction of trying to get more data there.
I think that would be incredible. Some of the fragmentation techniques that don't cause scrambling.
Yeah, UVPD. Sorry.
No, go ahead.
I was just going to say UVPD is obviously paving a way there. Something that's very hot.
Yeah, I think it's a fantastic field to get into. I mean, I think that there's actually, it's not slowing down. I think it's taking off. I think it's going to continue to do that as we rely on the precision of the measurements.
Yeah, it's definitely a romancing idea that an old technique is really getting a rebirth right now. And I think that's a great a great sentiment to finish on. So yeah, I just want to say thank you very much for an excellent talk, Ben. This concludes the presentation, everyone. So thank you very much for joining us. If you'd like to speak further about HDX, any of the topics that you saw today, please feel free to reach out to myself or Trajan, or I'm sure Ben would be very happy to have you guys contact Ben directly. Is that correct?
Sure, absolutely. Yeah, I think my email was on the opening slide, which doesn't do us much good now, but it'll be posted.
The recording will be out. But if you want to reach out to Ben, you can find him, or you can reach out to me directly. So yeah, thank you very much, Ben. Thank you very much, everyone, for joining us. All that's left to say is thank you and have an amazing day, everyone.